in clinical trials, everything ultimately comes down to the quality and integrity of data. Regulators approve drugs based on whether the clinical data is trustworthy. We have to see if it's reliable, consistent, and traceable, and whether it was generated in accordance with the pre specified approved protocol. And what you must know is clinical research professionals work in environments where those regulatory expectations must be met in real time across multiple sites, teams, and systems, often under tight timelines and operational constraints. And clinical trial protocol plays a major role in setting the direction for the process. The teams developing clinical trial protocols work towards creating a document that provides a clear executable framework for trial conduct. In this video, we'll dive in to understand what a clinical trial protocol really is, what it contains, and why it plays such a critical role in clinical research and drug development. What is a clinical trial protocol? Clinical trial protocol is the blueprint of clinical trials, tells investigators exactly how the study must be conducted without ambiguity. Now, think of it like an architectural plan. If the plan is unclear, the building will have cracks. But in clinical research, the stakes are far higher. Once a clinical trial is approved, this protocol is distributed across multiple sites, investigators, monitors, ethics committees, regulators, often across countries and different time zones. It becomes the primary document that aligns everyone on what must be done, when it must be done, and how deviations will be handled. Now, a protocol is highly specified and operational document. It includes first, scientific rationale, why this study is being done and what gap it is addressing. Second, study's objectives, what the trial is designed to prove or evaluate. Third, study design, phase randomization, blinding, control arms. Fourth is methodology, how procedures will be executed at the site level. Fifth is statistical considerations, sample size, logic of analysis plan. Sixth is inclusion and exclusion criteria, who can and cannot participate. Seventh is the endpoints, what outcomes define success or failure. Eighth is the treatment procedure, dosing, administration, visit schedules. Ninth is the safety monitoring plans, how adverse events are tracked and how they are reported. 10th is the data collection and handling, what data captured, when, and how. Every line in this document has operational consequences. Now, let us look at the intent of the clinical trial protocol. The protocol standardizes the process with a clear intention to preserve data integrity, maintain statistical validity, ensure consistency across trial sites, minimize protocol deviations, enable regulatory confidence in the data. Without this standardization, the trial results simply won't stand scrutiny. So, who actually develops clinical trial protocols? Protocols are developed by sponsoring organizations, often in collaboration with CROs. This is never one person's job. It is deeply collaborative process. It involves multiple stakeholders, clinical investigators, medical writers, biostatisticians, safety and pharmacovigilance experts, regulatory affairs specialists, CRO operations leaders, feasibility experts or patient representatives. Each role contributes to a different lens. Now, how do clinical trial protocols work in real world? Once a clinical trial begins, it operates in an environment where uncertainty is inevitable. Scientific assumptions are tested in real patients. Operational realities emerge across sites. New information surfaces as the study progresses. Each of these moments forces the team to stop and ask hard practical questions. Does this affect how we interpret the data already collected? Are sites still executing the protocol the same way, or are variations creeping in? Do we need to clarify instructions, tighten controls, or formally amend the protocol? And if we change something, what happens to the validity of the study? These decisions cannot wait until the end of the trial. They have to be made while patients are enrolling, data is flowing, and oversight is ongoing. And when those decisions are made, they directly act on the protocol. They determine whether the protocol needs clarification to prevent inconsistent execution. Certain procedures need tighter definitions to protect the data integrity. Deviations can be managed within the existing framework, or a formal protocol amendment is required. In fact, a study published on PubMed shows that as many as 57% of clinical trials undergo at least one formal amendment after they begin, and many have multiple changes over the course of study. Therefore, clinical trial protocols are designed to anticipate risk, control deviation, and preserve data integrity even as change occurs. Creating a clinical trial protocol requires scientific understanding, regulatory awareness, and operational thinking. That's why it's typically handled by experts with hands on trial experience and cross functional exposure, professionals who can anticipate real world challenges. If you are a life science graduate or a part of ecosystem that supports professionals in building real world job ready skills, subscribe to our channel and hit the bell icon to stay updated with educational content and industry insights across life science careers. Thank you for watching.
Visit us today, www.clinilaunchresearch.in.